Good morning, students. Myself, Major Komal Katyal, Department of Zoology, GK Gowani Government College, Pinmal. In continuation of my lecture on embryology, today I will discuss about cleavage. आज हम बात करेंगे विदलन के बारे में. What is cleavage? विदलन क्या है? Cleavage is a rapid series of mitotic divisions that occur just after fertilization. विदलन एक प्रक्रिया है जिसके दौरान समसूत्री विभाजन बहुत तीव्र गति से होता है और ये सारी प्रक्रिया निषेचन के तुरंत पश्चात होती है. There are two critical reasons why cleavage is so important. Number one, generation of a large number of cells that can undergo differentiation and gastrulation to form organs. पहली जो इम्पोर्टेंस है इसकी वो यह है कि बहुत अधिक संख्या के अंदर कोशिकाएं बनती हैं जिनमें विभेदन होता है और फिर गैस्ट्रुला भवन बनता है जिसके पश्चात अंग जनन की प्रक्रिया प्रारंभ होती है. The second is increase in nucleus cytoplasmic ratio. Eggs need a lot of cytoplasm to support embryogenesis, and it is difficult or impossible for one nucleus to support a huge cytoplasm, and oocytes are one of the largest cells that exist. One small nucleus just cannot transcribe enough RNA to meet the needs of a huge cytoplasm. दूसरी जो समस्या आती है वो ये होती है केंद्रक और कोशिका द्रव्य के बीच में जो अनुपात है. चूंकि जो डेवलपिंग oocyte है जिसके अंदर एम्ब्रियोजेनेसिस हो रहा है उसके अंदर कोशिका द्रव्य की मात्रा बहुत अधिक होती है और एक केंद्रक द्वारा उसका नियंत्रण करना संभव नहीं हो पाता. इसलिए बहुत तीव्र गति से इसमें समसूत्री विभाजन होता है बिना कोशिका की आकृति या आकार में वृद्धि हुए बिना. Now, let us therefore a larger nucleus to cytoplasmic ratio is optimal for cell function. So cell division occurs rapidly after fertilization to correct this problem. Now, what are the difference between cleavage and mitosis? That is the difference between cleavage and normal mitosis. Number one, blastomeres do not grow in size between successive cell divisions as they do in most cells. This leads to a rapid increase in nucleus cytoplasmic ratio. It means number of nuclei are increasing gradually while size of cells are decreasing gradually. It means the percentage of nucleus is increasing in proportion to the cytoplasm. Cells undergoing cleavage have mainly S, that is the synthetic phase, and M, that is the mitotic phase, while very little or no pre-mitotic or post-mitotic gap means they have a very short G1 and G2. Cleavage occurs very rapidly and mitosis and cytokinesis in each round of cell division are complete within an hour. Typical somatic cell divide much more slowly, several hours to days, and even the fastest cancer cell divide much slower than occurs in a zygote during cleavage. This is very clear from this example कि कैंसर की कोशिकाओं में भी जो कोशिका विभाजन की दर होती है उससे बहुत अधिक या उससे बहुत ज़्यादा तीव्र गति से विदलन के अंदर कोशिका विभाजन होता है. Cleavage differs in different types of eggs. The presence of large amount of yolk alters the cleavage patterns, leading to incomplete cleavage that characterizes birds and reptiles. Now, two areas of interest as far as cleavage is concerned. One, how does the process of cleavage differs in different organisms? And second, what mechanism regulate cleavage? So we were talking about two areas of interest in cleavage. This is the slide showing normal cell cycle with mitosis, the G1 S and G2 phase, and there is a long interphase as you can see in the normal cell cycle. While if we look at the mid-blastula transition, you can see the number of cells per embryo and the time taking in cell division at 15 degrees Celsius, beginning from cleavage to blastulation, gastrulation, neurula, that is a neurulation, tail bud formation, and ultimate tadpole. So here we are considering the example of a frog. In frog, generally it takes 1 hour to complete a cell division during cleavage. If we consider another example, that is of goldfish, it takes only 20 minutes to complete a cell division, while in case of mammalian example, the mouse take approximately 10 to 12 hours to complete one cell division of a cleavage. This is again slide showing the different phases of the mitotic cycle, beginning from the interphase, prophase, metaphase, anaphase, and telophase. From prophase to telophase, the M part or the M phase, the mitotic cycle, and the last one, the cytokinesis. So complete cell cycle include the karyokinesis, that is the division of nucleus, and then the cytokinesis, that is the division of cytoplasm. Now again, after the formation of two daughter cells, the actin filaments which form the contractile which helps to cleave the cell, and you can see here two daughter cells are formed. Now this is how the mitotic spindle is taking place. The black color X-shaped chromosomes are at the metaphasic plate, and spindle fibers plus centriole and kinetochore are shown in the slide. Now let us summarize the embryonic cleavage. It is the rapid division to produce many cells. Second, it increases the nucleus to cytoplasmic ratio from 1:550 to 1:6. There is distribution of cytoplasmic determinants. No growth occurs during cleavage. This is very important. विदलन के दौरान कोशिका के आकार में कोई भी वृद्धि नहीं होती है. The volume and general shape of the embryo does not change during cleavage. The resultant cells are known as blastomeres. Pace of cleavage is determined by the cytoplasm, unlike the normal mitosis where the pace of cell division is determined by the nucleus. Initial induction or differentiation for gastrulation occurs in the cleavage. Initial axis formation to maintain proper polarity during further embryonic development. As I've already told you, during oogenesis that there is clear-cut axis between animal pole to vegetal pole. Depending upon the intermitotic interval, it can be divisible into three phases: the early cleavage, the late cleavage, and the terminal cleavage or the terminal phase. Now let us discuss about the various planes of cleavage. The first one is the meridional plane. The meridional plane, when the furrow passes through the polar axis of the egg and bisect both the animal and vegetal pole. Second one is the vertical plane. The vertical cleavage furrow passes through the animal and the vegetal pole, but not through the median axis. The third one is the equatorial plane. The cleavage furrow is horizontal and is laid down in the equatorial at the right angle of the main axis between the animal and the vegetal pole. Then the last one, the latitudinal plane. The cleavage furrow of the latitudinal division are laid down transversely or horizontally, but not on the equator. For example, the third cleavage planes in the egg of Amphioxus. Now let us discuss about the cleavage patterns. What do you mean by cleavage patterns? Cleavage pattern, it means the different type of planes of cleavage follow a definite pattern, and these are known as cleavage pattern. The yoke or distribution of yoke and amount of yoke affects the cleavage patterns. Say for example, alacetal and isolacetal egg, it is holoblastic, जिनके अंदर योक या पीतक की मात्रा बहुत कम होती है, उस तरह के अंडों में पूर्ण भंजी विदलन होता है. In the case of centrolacetal and telolacetal egg, it is miroblastic, जैसा कि पक्षी, सरीसृप और कीटों के अंदर होता है, जिसको बोलते हैं हम अंशभंजी. The orientation of mitotic spindle. Categories are generally radial, bilateral, rotational, superficial, and spiral. Now the two type, the determinate cleavage and indeterminate cleavage, मतलब निर्धारी और अनिर्धारी विदलन. In the case of determinate cleavage, development state of each embryonic cell is fixed very early, characteristic of mollusk, annelids, and arthropod. If the first 4 cells of a snail embryo were separated, they only develop into a fixed 1/4 of the larva. Indeterminate cleavage, अनिर्धारी विदलन. Each embryonic cell's fate is not fixed. Each cell capable of forming a complete, although a small individual. Characteristic of higher non-chordates like echinoderms and chordate. Now let us take the example first of sea urchin, where there is radial and holoblastic अरीय व पूर्णभंजी विदलन shown through the diagram. After the sperm contact, there is presence of male and female pronuclei, formation of zygote, and various planes are shown up to the formation of blastocyst. This is the photograph of isolacetal egg. You can see the zygote, the 2-cell stage, the 4-cell stage, and the 8-cell stage. The cleavage is holoblastic, radial, मतलब पूर्णभंजी व अरीय विदलन. Let us take further the morula stage and the blastula stage of the isolacetal egg. Now the second example, that is of amphibian, whether it is bilateral or radial, it is holoblastic. You can see there is presence of animal pole, the gray crescent area, the vegetal pole, and ultimately you can see there is formation of a blastocyst, कोरक गुहा का निर्माण. This is what you can see in the case of an amphibian embryo. After the formation of blastula, there is formation of germ layer. It means the gastrulation is taking place with the formation of 3 germ layer like endoderm, mesoderm, and ectoderm. So this is about the 2 examples which we have studied today. First the sea urchin, and the second one is of frog. Thank you. This finishes my lecture part 1 on cleavage. इसी के साथ cleavage का भाग प्रथम समाप्त होता है, जिसमें हमने cleavage के पैटर्न्स, प्लेन्स और cleavage किस प्रकार से सामान्य समसूत्री विभाजन से अलग है, इसकी क्या खासियत है, किस प्रकार से अनिर्धारी और निर्धारी विदलन एक दूसरे से विभेदित होते हैं. थैंक यू.